Here we have our little buddy, the chloroplast. Chloroplast is made of two parts, the thylakoid and the stroma, the two locations of photosynthesis. The light reactions take place in the thylakoids, while the Calvin cycle takes place in the stroma. Let's dive into our light reactions. The red molecules represent our oxygens. The orange molecules represent our hydrogens. The blue molecules represent our carbons. First, water is split into hydrogen ions and oxygen, releasing O2. Electrons are taken from water and passed through photosystem 2. The electrons are then passed through the electron transfer chain in photosystem 1 by electron carriers that become increasingly electronegative, creating a hydrogen concentration gradient used by ATP synthase to make ATP, which is used in the Calvin cycle. The electrons are used to make NADPH, which is also used in the Calvin cycle. Now for the Calvin cycle. The three phases are carbon fixation, reduction, and regeneration. First, the carbons of three carbon dioxide molecules are taken and transferred by the enzyme Rubisco to PGA, a three-carbon sugar. This is called carbon fixation. Next, ATP and NADPH, the sources of energy for carbon fixation, are transferred back to ADP and NADP+. PGA is then transferred to PGAL. This is reduction. PGAL can be used by the cell to produce glucose. Three more ATP molecules are used to regenerate ribulose biphosphate, a 5-carbon sugar that will be used in the Calvin cycle again. During the light reactions, we already discussed linear light reactions. Now we will go over cyclic light reactions. Instead of using the electron in photosystem 1 to regenerate NADPH, the electron is transferred back to photosystem 2. In photorespiration, the stomata of plants close to conserve water. The problem with this is no CO2 can come in and the concentration of oxygen increases. Oxygen is used to make a 2 and 3 carbon sugar that is useless to the cell. This process is useless because the cell wasted ATP instead of making glucose.